Good morning, everyone. I hope you are all being safe and healthy and you enjoyed your spring break. Well, we are back to our lectures now. I don't know when I will be able to see you next, but as long as we are online, I will keep sending you the class lectures, the video recordings, and we will have our scheduled quizzes and tests as well. Just another announcement that I will not be holding office hours in person. So if you have a question, if you want to talk, the best way to reach me is the email through Web Campus or directly to my email address. All right, with that said, let's move on to our topic of discussion today. Before spring break, we were talking about DNA replication, and we will be continuing that topic today. And then we will move on to our next module on DNA repair. Okay. So before spring break, we were talking about DNA polymerases, that all DNA polymerases have the similar structure, right-hand like structure. DNA polymerases are composed of multiple different subunits together making a holoenzyme. 3 DNA polymerases work together for polymerization of DNA for adding new nucleotide during DNA replication. The core of the core enzyme of this holoenzyme consists of 3 subunits, alpha, epsilon, and theta. Here you can see in this figure, in this image, this largest part of this part right here is the core enzyme, core polymerase. It consists of 3 subunits. This alpha subunit is the polymerase subunit. That's what that has that polymerization activity of this enzyme. Epsilon subunit has a 3 to 5 prime exonuclease activity, so this is important for proofreading. If there is a wrong nucleotide added, the DNA strand will move to this epsilon subunit, and that nucleotide will be removed. Yes, epsilon. Theta subunit activates this proofreading activity of epsilon, because this doesn't have to be activated all the time, because since DNA is not polymerase is not going to make too many errors, so it's only get activated when there is an error. Okay, next is these beta clamps. These beta clamps structure here, these are associated with the holoenzyme, and their function is to hold on to DNA molecule when the polymerization is happening through this core polymerase. Next part is tau. Tau keeps these holoenzymes attached to the gamma subunit. So it acts as it's a dimerizes or trimerizes the core enzymes, because now we know that there are 3 instead of 2. Gamma unit acts as a clamp loader, so the major function of gamma subunit is to load these clamps onto the DNA molecule. The sky here is quite important subunit of your holoenzyme as well, because this mediates the switch from RNA primer to DNA. Remember, DNA polymerase cannot act de novo, so it requires an existing hydroxyl group, and that hydroxyl group is provided by this RNA primer. Okay. We talked about the mechanism in detail before spring break, how the pre-primosome complex is formed and how the primosome complex is formed. This primosome complex consists of primase or DNA G in E. coli and helicase or DNA B in E. coli. These associate together to form primosome. Primosome really means that synthesis of short RNA. Short RNA primers onto DNA molecule. Once these RNA primers are formed, the primosome is released, although helicase still stay associated with it will associate with the polymerase. As I mentioned, helicase, the activity of, or I should say processivity of helicase depends on the presence of DNA pol 3. When the helicase is associated with DNA pol 3, so here's helicase, this hexamer, this ring-like structure, and if this is associated with the DNA polymerase, this is a processive enzyme. That means it can catalyze multiple reaction once it's bind to the molecule. So that means it can open a large chunk of DNA for synthesis. These proteins are single-stranded DNA binding protein. These will attach immediately when the helicase opens the DNA, so the DNA is not exposed. In case helicase is not associated with the DNA pol 3, then it's not a processive enzyme anymore, so it opens only one or two nucleotide at a time. It will not catalyze multiple reaction with one binding. So it's really important for DNA helicase to be associated with DNA polymerase for DNA replication to occur at speed. Topoisomerase is another important enzyme. As you all know now, as the DNA strands are opening, it puts a strain on the DNA ahead of replication fork. So this right here is the replication fork, and as it opens, it will make the DNA ahead of it positively supercoiled. Positively supercoiled DNA is hard to open. Therefore, topoisomerase makes the break in the DNA strand, and it will release that strain to convert it into a negatively supercoiled DNA. Okay, so now we talked about all this on Wednesday, but just to remind you, this tau is right here. Tau binds to both the core, which is here in green. These are the core enzyme, the three units of core, 1, 2, and 3. This tau binds to both the core enzyme as well as the gamma complex. The gamma complex is right here. It's a trimer. So 1, 2, and 3 bound to 1, 2, and 3 of these core enzymes. Gamma complex, as I already mentioned, is function as a DNA-dependent ATPase. It uses the ATP energy from ATP, and the most important function of gamma complex that appears to be is to load this beta clamp onto the DNA molecule. Here, all these in green shows the RNA primer. So this is the leading strand. These are the lagging strands. Each of these Okazaki fragment has its own RNA primer here. This is already synthesizing, so it will, this here there's a newly formed RNA primer. So this gamma complex will take this sequence, will put this beta clamp onto that sequence. Once the beta clamp is formed, the DNA polymerase will move along with this beta clamp and will synthesize this strand. So here, once the one of the Okazaki fragment that is synthesizing reaches to the next RNA primer that's already been synthesized, this is the affinity of beta clamp to the polymerase will be less, and it slides off, or it's removed from here. Although it doesn't go far, because this is still part of holoenzyme, so it's still attached. So this will, the gamma complex will grab it and put it on the next sequence. Okay. This video talks about whatever we talked about in detail. So if you want to watch this video, this is pretty nice. Shows how the beta clamps are being put on by these gamma complex and the helicase activity as well. Now, we talked about DNA. We've been talking about DNA replication, really focusing on initiation and elongation, because the replication happens in 3 distinct steps, right? So initiation, that's where all the primosome activity occurs, and elongation is when the holoenzyme is bound to DNA and it starts adding these nucleotide to polymerize the DNA. That's the elongation. Now comes the termination, the last step in DNA replication, but equally important. So here is the example of a prokaryotic DNA replication. As you remember, prokaryotic DNA is circular. So this circular DNA, replication of circular DNA is a little different than the linear eukaryotic DNA. You also probably recall that in prokaryotes there is only one origin of replication. So this is the origin of replication, and that's where replication will initiate. Replication occur in is bidirectional, so it occurs in both direction, clockwise as well as counterclockwise. So there's one replication fork that's moving in clockwise direction called clockwise fork. The another one is moving counterclockwise. It's called counterclockwise fork. Termination happens at these specific sequences called ter sequences or termination sequences. There are two different sets of termination sequences, group 1 and group 2. These restricted terminus region or these ter sequences, these are combination of a DNA motif, which is about 20 to 30 base pair, and they also have a cognate termination protein that recognizes these ter sites, which is called a TUS protein. This TUS protein binds to these ter sequences and will inhibit the movement, or it will not allow the fork to pass through. So counterclockwise fork can pass through group 2 ter sites, but it will not be able to pass through group 1 site, so it will stop there. Similarly, the clockwise fork will be able to pass through group 1 but not through group 2. So that way it will just halt, the replication will halt here opposite to the origin of replication. Okay. So this diagram here shows it a little better, where one of this color blue is the parental strand and purple is the newly synthesized strand. Here the fork is moving, the fork moving in this direction will be stopped at the ter 1 site or group 1 terminal regions, and the other one, that's the fork that's moving the other direction, will stop here at ter 2. This is the TUS protein shown here is bound to ter region. Ter DNA, these are DNA binding proteins, so they are bound to these ter sequences. They will not allow this fork to pass. So it prevents the advancement of fork moving in either direction, one or the other direction, depending on which way the replication fork is moving. Okay. Now you remember, or you can see right here, it's a pretty good example here, as when this the circular DNA is replicating, it ends up being joined together at this terminal site, or this the DNA, or as it's called, it's catenated DNA at this point. So this catenated DNA need to be decatenated in order for these two molecules to separate. So here, this is, if you look at it, it's because it's all double-stranded, circular double-stranded DNA. This is the origin of replication. That's where it starts. The replication starts, and that once the replication is complete, this will be bound together or attached together. In order for these two to separate, the topoisomerase activity needs to be there. This topoisomerase activity, which is topo 4 in E. coli, this makes a double-stranded DNA cut, and this double-stranded DNA cut then is sealed by ligases. And now there are two different molecules. This is the most we know about termination of DNA replication. It is not very well understood, so there's still a lot of work going on to completely understand DNA replication termination. So now that's what we talked about as a prokaryotic DNA molecule, so that is circular. But eukaryotic DNA molecules are linear DNA molecules, and then there's a problem of how to replicate the ends of the chromosomes, right? What happens in eukaryotic, in a linear chromosome, is, since we know that one of the leading strand is continuously replicated, but the lagging strand is not continuous, is discontinuous. So each of these Okazaki fragment have their own RNA primer. These in green are the RNA primers. So here, with the lagging strand, there is no problem. It's completely synthesized. But with the lagging strand, this last fragment is hard to form, because this is the RNA primer, and once you remove this RNA primer, there is no other sequence to attach to. And what happens in that case is exonuclease, because RNA primers is removed, but there's nothing to complete it. So the exonuclease activity will cut this portion of the DNA, this portion of the chromosome, and since they both chromosome need to be similar length, as all of this will be chewed by the exonuclease, and now the chromosome is shorter than the parental chromosome. So that's why we have telomeres on the linear chromosomes, because the telomere prevent this end of the shortening of the eukaryotic chromosomes, because those are repeat sequences, and these repeat sequences do not code for anything. Okay, so here the telomere replication. Telomeres are ends of the linear chromosomes. These are repetitive sequences. They do not code for anything, because their main important function is to avoid the shortening of DNA during replication. In human, telomere is a 6-base pair sequence, TTAGGG. It's repeated thousands of times, and after each DNA replication, some of the telomere sequences get lost, but it doesn't really impact, because these do not code for anything. Polymerase enzyme that catalyzes the replication of telomeres. It's a special enzyme specifically for telomere synthesis or telomere replication. It's a terminal transferase, or it's a reverse transcriptase. It carries an RNA molecule with it, and the sequence of that RNA molecule is CCC-AAU-CCC. This is used as a template, and when it elongates the telomere. The telomere, they add a complementary RNA base at the 3' of the DNA strand, and this is its complementary base. And you will see that in next slide, it loops around the telomere, so it's always available for the next synthesis reaction. In this slide, you will see that this telomere binds to 3' overhang. Telomerase binds to 3' overhang of the telomere. So this is your telomere, 3'. This binds here, and then it starts adding the complementary sequences. Once it adds this sequence, then there is so this is available now for the polymerase to fill in this gap, because that's that little gap where the RNA primer was in the Okazaki fragment that it was not able to add on to. So here, so here, once the telomerase starts adding those sequences, then the polymerase comes in and it extends that portion where the RNA primer was and put this complementary sequence again, so it does not lose any of the information in the DNA in the chromosome. So here it also it shows in a little better way what that 3' loop looks like. So this is your chromosome, and here this is an additional portion, this RNA primer of the telomerase that just loops around. This is a single-stranded 3' end which loops around to itself to make this double-stranded molecule here. One of the things that you want to keep in mind, that telomerases are not active in most somatic cells, so that will be most of our cells, our body cells. And it's active in germ cells, because those germ cells make sperms and eggs, so they have to be activated because they divide more often than the somatic cells. And they're also active in adult stem cells, because all of these cells, they go through many divisions. Cancer cells, however, they have the activated telomerase, and that's how they are able to divide multiple times more than the regular cells. That's why they are immortal. There are many drugs that use these telomerase inhibition as a part of cancer therapy to stop the telomerase activity in cancer cells.